Welcome to a Rice University digital media production. For more information about us, please visit our website at www.rice.edu.
Good afternoon. Welcome to today's lecture in Sciencia's year-long series on networks. I'm Susan McIntosh, and I'm the director of Scientia. So far this year, our speakers have explored social networks, network models for H1N1 viral transmission, knowledge networks, infrastructure networks, and the general properties of scale-free networks. Today, we turn to what must surely be the most complex of all networks, neural networks in the human brain. Many of you may remember Nobel laureate Eric Kandel's marvelous Bachner lecture in our memory series 2 years ago, which highlighted some of the basic molecular mechanisms that underlie the storage of experience as memory. This was some of the basic path-breaking work that led not only to new understandings of the individual cells involved in information processing, but also to further research that illuminated the network architecture of which they are a part. Our lecturer today did his thesis research in the laboratory of Dr. Kandel at New York University Medical School, and then spent an additional 2 years as a postdoctoral fellow with Dr. Kandel at the College of Physicians and Surgeons of Columbia University. He then assumed a faculty position in the Department of Physiology at the University of Pittsburgh School of Medicine. In 1982, he moved to the Department of Physiology and Cell Biology of the University of Texas Medical School at Houston, and in 1987 was appointed professor and chair of the Department of Neurobiology and Anatomy. In addition to his primary appointment, Dr. Byrne holds positions as adjunct professor in the Department of Psychology and the Department of Electrical and Computer Engineering here at Rice. Dr. Byrne's research interests are in the neuronal and molecular mechanisms underlying simple forms of learning. Molecular, biochemical, biophysical, electrophysiological, and imaging techniques are used to analyze the properties of the neural circuits mediating the behaviors, the individual neurons comprising those circuits, and the biochemical and gene networks within those neurons. The empirical analyses are complemented with realistic mathematical modeling in order to determine the extent to which observed processes and their interactions are sufficient to explain the behaviors and their modifications. His publications include around 200 refereed and invited journal articles and 7 co-authored or edited books, 2 of which are in second editions. Among these are Learning and Memory, published by Macmillan, and From Molecules to Networks, An Introduction to Cellular and Molecular Neuroscience, the second edition of which was published by Elsevier in 2009. Dr. Byrne serves or has served on the editorial boards of over a dozen journals of neurobiology, physiology, and memory, and has been editor-in-chief of Learning and Memory since 1996. If his research output is prodigious, it is a reflection of his phenomenal grantsmanship. He has attracted over $20 million in NIH funding as PI over the last 10 years. His research accomplishments have been recognized by his receipt of numerous honors and awards, including a National Institute of Mental Health Research Scientist Award, the Jacob Javits Neuroscience Investigator Award from the National Institutes of Health, and the Hebb Award from the International Neural Network Society. In 1999, Dr. Byrne received the Presidential Scholar Award, the highest academic honor bestowed upon a faculty member at the University of Texas, Houston. In 2006, he received the UT Houston's President Award for Mentoring Women. Dr. Byrne was elected as a fellow of the American Association for the Advancement of Science in 2002. We are indeed fortunate to have Jack Byrne with us today to speak on neural networks. Please join me in welcoming him to the podium.
Thank you very much. Well, thank you very much, Susan, for that wonderful introduction. It is indeed a great pleasure and honor to be here today and to participate in this wonderful session that you've been organizing for the past several years. I very much hope that my talk lives up to the quality of some of the previous ones that I've been to and also fits in nicely with this theme on networks. Well, as Susan indicated, today I'm going to talk about the networks that are found within this 3 pounds of jelly-like material found within our skulls. Now, in this landscape are found love, charity, hope, Beethoven's Fifth, Apollo 11, Hamlet, and the pyramids. So here are clearly the guts of the most complex machine in the world. It's the organ of the past and the master of the future, the dreamer of dreams and the heart and soul of life possessed by every single one of us. Now, all these phenomenal features of the human brain would not be possible without the hundreds of billions of neurons that make it, and most importantly, the connections between those neurons. Now, I think it's fair to say that we're just beginning to scratch the surface in understanding the brain and how it works. But neuroscience over the past 40 years has made great strides in understanding this complex network. And what I want to do today is talk about several aspects of the brain and how it works and how the networks are used, if I can advance the slides. I first want to talk about the neuron. It's the elementary node or element of the brain, and really you can't understand neural networks unless you understand the basic properties of the nerve cell. Now, what makes the nervous system such a fantastic device is not only that it has 100 billion neurons, the liver probably has 100 billion liver cells, but what distinguishes the brain from other organs of the body is that the nerve cells are capable of communicating or connecting to each other. And there are 2 flavors to these types of connections. Some neurons excite each other, these are called excitatory connections, and other neurons inhibit each other. So in order to understand neural networks, you need to understand the nature of the ways in which one cell communicates with another cell. From that, we can begin to understand some simple, what I'll call micro networks. Networks that generate or mediate reflexes, networks that are involved in processing sensory information such as contrast enhancement in the visual system, networks that are involved in generating locomotion. Then talk about a so-called nano network that generates circadian rhythms. I call this a nano network because it doesn't really involve a neural network per se, rather it involves a biochemical genetic network within neurons. Then talk about an issue dear to my heart, and that is the networks that underlie learning and memory. And finally conclude, albeit briefly, with a discussion of macro networks, which are networks that are involved in more complex, higher order functions of the brain, such as cognition. Now, the key player here is the nerve cell. There are 100 billion or so of these in the brain, but they all share a number of common features. First of all, neurons are distinct from most other cells in the body in that they are polarized. There is a region of the neuron, the so-called receiving end, if you will. Morphologically, these are known as dendrites. It's on these branches where one neuron receives connections from another neuron. Then we have the cell body, or soma. That's where the nucleus is found and the other organelles necessary for cellular function. A key component of nerve cells is this long axon. This can be very long, up to a meter or so in length, and it's over the nerve axon where information is transmitted from one part of the neuron, say the cell body, to the terminal regions of the neuron. Now, these terminal regions, also called synapses, are where all the action takes place, because it's at the synapse where one neuron releases a chemical neurotransmitter substance, allowing it to communicate with another neuron, like the blue cell shown here. So the blue cell would be referred to as the postsynaptic cell, this other cell as the presynaptic cell. So one neuron makes a contact through these synapses with a postsynaptic cell. And one neuron in turn receives contacts from many different other neurons, fortunately color-coded here. So you see 3 neurons contacting this one postsynaptic neuron. Now, the types of connectivity can be very, very rich. It's been estimated that one neuron can contact not just a single postsynaptic cell, but up to 10,000 other postsynaptic cells. And in turn, one neuron, like this one, can receive contacts or input from 10,000 other cells. So the potential complexity for the networks is very, very vast. Now, it's important to understand what happens at the synapse. How does this chemical communication take place? Well, here's a blow-up view of the synapse. Within the synapse are neurotransmitter containing vesicles. So these little blue dots represent the neurotransmitter. The brown circle is the vesicle. So these vesicles contain neurotransmitter. When a nerve impulse invades the synaptic terminal, it causes the fusion of these vesicles with the membrane, causing the transmitter to be released through a process called exocytosis. The transmitter then diffuses across this gap between the pre and the postsynaptic cell. Note the presynaptic cell is not directly connected to the postsynaptic cell. So in order to communicate with the postsynaptic cell, it needs to release this transmitter. The transmitter diffuses across the cleft very rapidly, where it combines or interacts with receptors on the postsynaptic membrane. Those receptors lead to permeability changes and in turn potential changes in the postsynaptic cell. So we're going through a little bit of Neurobiology 101 to introduce the network topics. So one of the critical features about nerve cells is, in order to understand their operation, you need to understand a little bit about how they work, and they work through electrical signaling mechanisms. So here's an example of an idealized nerve cell, and in the extracellular medium is placed a microelectrode. Microelectrode is nothing more than a small piece of capillary tubing that's stretched under heat to produce a very fine tip. It's filled with a conducting solution and then connected to a suitable recording device like this oscilloscope. With the electrode outside the cell in the extracellular medium, there's zero potential because the extracellular medium is isopotential. If, however, you take this electrode and penetrate the cell with the electrode such that the tip of the electrode is now inside the cell, you see a sharp deflection on the recording device, and you now record a potential of minus 60 millivolts, inside negative with respect to the outside. This is the so-called resting potential, and it's constant for indefinite periods of time in the absence of any stimulation. If you remove the electrode, you again record the zero potential. Now, resting potentials are not just characteristics of nerve cells. All cells in the body have resting potentials. But what distinguishes nerve cells and other excitable membranes like muscle cells is that they're capable of changing their resting potential for, in the case of nerve cells, for integrating information and transmitting information, and in the case of muscle cells, for producing contractions. So let me just briefly introduce you to the nerve action potential or nerve impulse. So here is another simple schematic diagram of an idealized neuron. This cell has been impaled with one neuron to measure the resting potential, but it's also been impaled with a second electrode, which we'll call, for lack of a better term, the stimulating electrode. Now, the stimulating electrode, why it's called that will become clear in a moment, because the stimulating electrode is connected through this switch to a battery. Now, you can all imagine if you have the battery so oriented with the positive pole here, and you close the switch. As a result of closing the switch, you're going to make the inside of the cell somewhat more positive, depending upon the size of the battery, than it was before. So I'm just going to show you a little animation, and what's going to happen in this animation is that the switch is going to be repeatedly opened and closed, and each time it's opened the next, a larger battery will be applied to the circuit. Initially, there will be small potential changes, and they'll get bigger and bigger. Eventually, we'll see the initiation or triggering of the nerve action potential. So here's the small battery, a small so-called depolarization, a decrease in the polarized state of the membrane. Larger battery, still larger depolarization. And now we get to a certain point where that depolarization reaches a potential called the threshold. When this all or nothing new event is initiated, this is the nerve impulse or action potential. If you deliver yet a larger battery, you still only see a single action potential. The amplitude of the action potential does not change. So from this simple recording, you see two very important features of action potentials. One is that they're elicited in an all or nothing fashion. Either an action potential is elicited with stimuli at or above threshold, or an action potential is not elicited. Also, you see that action potentials are very brief events, only about several milliseconds in duration. The initiation of an action potential is somewhat similar to applying a match to a fuse. You need a certain temperature in order to ignite the fuse. That's the threshold. If you use a match that has a greater amount of heat, you'll still ignite the fuse. It won't burn any brighter or faster. So action potentials are elicited in all or none fashion, and also they are propagated in an all or none fashion. So once you initiate these action potentials in one portion of the cell, like the cell body, that action potential will propagate out the axon and ultimately invade the synapse, where it can initiate this process of synaptic transmission. Now, the next slide illustrates another feature of action potentials. Let me first point out that we only generated a single action potential here because the duration of the stimulus was so brief that there was only time to initiate a single action potential. But as shown in this panel of slides, if we make the stimulus intensity, if we make the stimulus a longer duration, instead of just 2 milliseconds, we make it a longer duration. With a weak battery, well, we still only initiate a single action potential. But if we do this again, now using a larger battery, there is a greater depolarization. And now, instead of eliciting one action potential, we get multiple action potentials. And with a yet larger battery, there is even more action potentials. So this indicates a third feature of action potentials, and it's the fact that the nervous system encodes information not in terms of the changes in the amplitude of an action potential, but rather in their frequency. So, and this is a very universal property, the greater the intensity of a mechanical stimulus to a touch receptor, the greater the number of action potentials that would be produced, the greater the amount of stretch to a muscle stretch receptor, the greater the intensity of a light, the greater will be the number of action potentials that are transmitted into the central nervous system. Similarly, in the motor system, the greater number of action potentials in a motor cell, the greater will be the contraction of the muscle. So it's frequency coding. Engineers would call this pulse frequency modulation. So that's the basic electrophysiology of nerve cells. They have resting potentials. If the resting potential gets to a certain threshold level, an action potential is elicited. The longer the duration of that stimulus, the greater the frequency of action potentials. Let's now return to this property of synaptic transmission. Now, there's two flavors to this, and the two flavors are illustrated in the next slide. Here we have two neurons. A green neuron that, for reasons that will become clear, we'll call the excitatory neuron, and that green neuron makes a connection to this blue postsynaptic neuron. Shown below is the consequences of initiating an action potential in the green neuron. That action potential leads to a change in the resting potential of the postsynaptic cell. You see the membrane potential changes from its resting potential, which would be minus 60 millivolts, to a more depolarized state. This is a so-called excitatory postsynaptic potential. It's excitatory because, as you can see, it tends to move the membrane potential towards the threshold. And it's a postsynaptic potential because it's a potential recorded on the postsynaptic side of the synapse. Now, generally, and this is an important point, a single action potential in a presynaptic cell, while it releases that chemical messenger to excite the postsynaptic cell, a single action potential generally doesn't produce enough excitation to fire an action potential. But if multiple action potentials are fired in the presynaptic cell, those excitatory potentials can summate, ultimately leading to the initiation of an action potential. So that's one flavor. That's the so-called excitatory transmission. And that's the way a presynaptic neuron can relay information to the postsynaptic neuron. If you will, this is the simplest neural network that you can envision, a network of just two neurons. Now, the next slide shows, and this, of course, gives a go signal to the postsynaptic cell. So we want to transmit information through this network pathway. The next slide shows what the consequences of adding to this simple circuit a third neuron. This one, for reasons that will be clear, this red one is going to be called an inhibitory neuron. So it also makes a synaptic contact with the postsynaptic neuron. It also releases chemical transmitter messenger, but the consequences of that messenger binding to receptors on the postsynaptic cell is exactly opposite to the consequences of this transmitter. And that's clearly shown here. So the red cell now shows a recording from the inhibitory neuron. Here is an action potential in the inhibitory neuron. And as you can see, the consequences of that action potential are to produce an increase in the resting potential. The resting potential is more negative than it was before. It is moving the resting potential away from threshold, and therefore this is called an inhibitory postsynaptic potential, right? Because it tends to prevent the postsynaptic neuron from firing an action potential. So this is a stop signal for the postsynaptic cell, right? This one's saying go, and this one is saying stop. Now, what's the neuron going to do? Well, what a neuron is doing is constantly adding up. It's like a little adding machine. It's adding up the summation of the excitatory and inhibitory synaptic input, and if that summation is at or above threshold, it fires an action potential. If it's below threshold, no action potential is initiated. And you can clearly see this with the next slide. So here, two action potentials are produced in the excitatory interneuron. Previously, those two action potentials led to a summated excitation, which fired an action potential. But if you fire an action potential in the inhibitory neuron just before those two action potentials in the postsynaptic, in the excitatory neuron, you still have the summation of the excitation, but now that summation is below threshold and no action potential is elicited in the postsynaptic cell. So the inhibitory neuron and inhibition in general is a way of gating or regulating the ability of an excitatory signal to fire a postsynaptic cell. So this is the most elementary neural circuit, just consisting of 2 or 3 cells in this case. And as I indicated, the complexity can be very, very vast because any one cell can receive 10,000 connections and make connections to 10,000 other cells. But despite that vast complexity, it seems as though there are a number of configurations that are repeatedly used by the nervous system. And some of those motifs are illustrated here. Don't worry about the complexity of this slide for the moment because I'm going to talk about most of these, again, individually. But I'll just give you a brief overview of the different kinds of motifs that one sees. And here we have these very common motifs. They don't involve thousands of cells. They involve just several cells, 2, 3, 4 cells. And we'll see that just connecting neurons together in these small micro networks can do some very interesting, mediate some very interesting behaviors and have some interesting computational capabilities. So part A is the feedforward excitation. This is the one I've already showed you. This is the way that one neuron can relay information to its neighbor. And you can have long chains of these so that information can be propagated through the nervous system. B is an interesting case. It's a feedback excitation. So a presynaptic neuron can excite a postsynaptic neuron. That postsynaptic neuron, in turn, can feed back and excite the presynaptic neuron. So I think you can see right away the functional role of this cell might be to serve as a switch-like mechanism, because once this presynaptic cell was activated, that activation could be recurrent and perpetuated. So activation here could switch this network on and could stay on. Then there's a phenomenon called lateral excitation. A presynaptic neuron excites its neighbors. Feedforward inhibition is an interesting network motif. A presynaptic cell excites an inhibitory interneuron, and that inhibitory interneuron then inhibits the next follower cell. So this is a way of shutting down excitation in a neural pathway. Part E is feedback inhibition. A presynaptic cell connects to a postsynaptic cell. The postsynaptic cell in turn connects to an interneuron, which then inhibits the presynaptic cell. So the functional consequences of this kind of circuit would be to limit excitation in the pathway. One would have some initial excitation, and then it would be shut off after this interneuron becomes active. Lateral inhibition, we'll be talking about that in more detail. A presynaptic cell excites inhibitory interneurons, and they feed back to inhibit the neighboring cells in the network. G is an extension of this feedback excitation. It's called recurrent excitation. A presynaptic neuron excites a postsynaptic neuron. That postsynaptic neuron can feed back to excite itself, or other neurons which ultimately feed back to itself. So once again, once this network is engaged, it could lead to a perpetuating and recurrent waves of activity. Here's an interesting neuron which seems to make a motif which seems to make little sense. One neuron connects to another to inhibit it. This neuron inhibits its neighbor, this neuron inhibits its neighbor, and this one inhibits the first one. It looks like this circuit would do nothing, but I hope to show you that this can produce some very interesting neural circuit behavior. And then there is convergence and divergence, which I've already mentioned. One postsynaptic cell receives convergent input from a number of different presynaptic cells, and any individual neuron can make divergent connections to many different postsynaptic cells. Okay, let's see some real-life examples of what these networks do. Let's talk first about feed-forward excitation and feed-forward inhibition. It turns out that these neural networks, the simple motif, is critical for mediating simple reflex behaviors, a reflex behavior that you all probably know about. It's the so-called knee-jerk or stretch reflex. You've all seen this. Maybe you've had it done to you. You go to a neurologist, takes their hammer, and they give a tap to the knee, and there's a subsequent extension of the leg, and this is used as a very crude way of testing the integrity of the nervous system. Well, this reflex, this simple behavior, is mediated by these feed-forward excitation and feed-forward inhibition networks. We just go through this slide step by step. So the hammer is delivered, and as a result of the hammer, there's a stretch of the muscle. That leads to the initiation of action potentials in sensory neurons. It's a little bright light there that represents an action potential. The action potential then is initiated in this all or none fashion. It then propagates. That's the little ball moving across this process. The action potential then these processes then go into the spinal cord, and there's two branches. Let me first talk about this branch. The action potential invades the synaptic terminal of the sensory neuron, causing the release of transmitter. The excitation and an action potential in the motor neuron. That action potential then propagates out the peripheral nerve to invade the synapse at the muscle, causing the release of transmitter, and an action potential in the muscle cell, producing the extension of the limb. So here you have a simple feed-forward excitation circuit that's mediating this behavior. Now let's look at this other limb here. Let me go back a little bit. So this sensory neuron has two branches, one which activated the blue neuron, and another branch which makes a synaptic connection to this black cell called an interneuron. It's called an interneuron because it's interposed between one neuron and another neuron. So again, we release the transmitter. It leads to an action potential in the interneuron. The interneuron then releases transmitter, but this time this transmitter is an inhibitory transmitter. So rather than exciting this flexor motor neuron, it inhibits the flexor motor neuron. And why is that important? Because it prevents or decreases the probability of this neuron being active and therefore having this muscle contract, the exact contraction that you don't want to occur. So this reflex is mediated by a combination of feed-forward excitation and feed-forward inhibition. Okay? Now, I misled you a little bit, because this circuit really wouldn't work unless there was something else going on. Because remember, I indicated that this neurotransmitter substance, which diffuses across the cleft and which is produced by a single action potential, produces an excitation of the postsynaptic cell, but that excitation is too small to fire an action potential. The way this circuit works is it takes advantage of convergence and divergence. There's just not one sensory neuron which is activated when you stretch the muscle. There are multiple sensory neurons, tens if not hundreds. And these sensory neurons then all project into the spinal cord, and they converge onto individual extensor motor neurons. So the combined effects of the multiple sensory neurons and their summated excitation then produces the action potential in the muscle cell, in the motor neuron. And there's just not one, so this is the convergence phenomenon, and there's just not one motor neuron. There are multiple neurons, so the sensory neuron diverges and connects to multiple motor neurons. So multiple motor neurons innervate multiple muscles. So the net effect when you stretch the muscle is that you activate multiple sensory neurons which converge onto individual neurons, and multiple motor neurons are active. So the reflex needs both feed-forward excitation, but also needs this divergence and convergence. Lateral inhibition is an interesting phenomenon that's very important in processing sensory information. So here are two bands, a dark gray band and a light gray band. Now, if you look at this carefully, you may note that the light gray band is a little bit lighter at the border of the dark gray band than it is in the other portions of the field. And the dark gray band is a little bit darker at the border than the other part of the dark gray band, even though this intensity, I should tell you, is uniform dark gray here and uniform light gray here. So this is a phenomenon of contrast enhancement, which allows the visual system to extract information from visual scenes. Now, this is mediated by a phenomenon called lateral inhibition, and that phenomenon occurs in the retina. Now, just to put this in perspective, let me show you the so-called network that mediates that without the lateral inhibition. So you have the light falling on the retina, and let's just say that the dark gray has a light intensity of 5, to make it simple, and the light gray has an intensity of 10. That light then activates these photoreceptors, and these photoreceptors then make synaptic connections to so-called second-order neurons, and then there's going to be some output. So let's just say that the light intensity of 5 leads to 5 action potentials per second, and the light intensity of 10 leads to 10 action potentials per second. So if there was nothing going on, the information that would be transmitted to the nervous system and what you would perceive would be exactly as the gradient of the intensity. But that's not what you perceive. What you perceive is that it's a little bit lighter here than it is here, and it's a little bit darker here than it is over here. And the way that works is through lateral inhibition. So each one of these photoreceptors is making inhibitory connections, synaptic connections to their neighbors. So the consequence over in this part of the field is that all of the cells are getting the same inhibition. They're getting the same excitation, and they're also getting the same inhibition. Over here, all the cells are also getting the same excitation, and they're getting the same inhibition, although weakly. But the key event occurs here, right at the border, because this particular neuron is receiving the same inhibition from this side of the field, but because this cell is responding to the dark, it's receiving less inhibition, so therefore it has more excitation. So this cell is responding with a higher frequency train of action potentials than the adjacent neurons. This neuron over here is receiving the same inhibition from its neighboring cells responding to the dark, but it's receiving more inhibition from the cell responding to the light. Therefore, its output is even less than its neighbors. So this simple circuit of lateral inhibition can account for this phenomenon of contrast enhancement. It can also account for a visual illusion, known as a Mach band. So here you see a gradient of light and dark bands, right? And across is a horizontal line. Now it appears as though this horizontal line has uneven distribution of intensities, with it being darker here and being lighter here. Does everybody see that? Okay, now, and I need to tell you that the intensity of this horizontal band is uniform throughout. Do you believe that? So let me show you by putting a little mask across there. Put a mask across there. I haven't changed this at all. I've just put a mask across there. Now you can see that this dark gray is uniform throughout. So the reason why it is darker here, is perceived as being darker, is because the cells in the retina that are responding to this light are being inhibited by the brighter field. This seems lighter because the cells are getting less inhibition from the cells responding to this dark region. So this phenomenon of lateral inhibition not only is important for contrast enhancement, but can help explain some so-called visual illusions. Now let me turn to this issue of how neurons can generate rhythms. And there's all kinds of rhythms that are physiologically important, ranging from our respiration rate, which occurs once every 20 minutes, once 20 times a minute or so, locomotor rhythms, and then there's the circadian rhythms that occur on a daily basis. Now, in one level, it's interesting that you don't need a network at all to generate a neuronal rhythm. In fact, an individual neuron is capable of an endogenous rhythm. So here's an example of a recording from a neuron that has this so-called endogenous bursting rhythm. It's actually called the parabolic burster because the nature of the interspike intervals being long here, and then they get very short, and then they get long again. So there's this burst of action potentials or impulses, then the cell becomes silent, and then there's another burst. It occurs about every 10 to 15 seconds, over and over and over again. This burst in this neuron actually occurs even if you were to surgically remove this neuron and place it in culture. So there's no synaptic connections, no network at all necessary for this rhythm. But it does involve a network. It involves what I'll call a nanonetwork of biochemical and biophysical processes. So here is just a schematic diagram of how this works. This would be the membrane, intracellular part of the cell, the extracellular part, the kinds of these little channels here are membrane channels that allow ions to flow. Critical for this neuron to work is an influx of calcium, which is represented by this green arrow. The influx of calcium leads to the burst of action potentials. But what happens is that as a result of the burst of action potentials and the influx of calcium, calcium levels within the cell accumulate. And what that calcium accumulation does is shut off the influx of calcium. So you have a burst, and then it gets shut off. The burst leads to influx of calcium, and then the burst gets shut off by that calcium. But there is a second burst. Why is there a second burst? Because that calcium is either buffered away or removed from the cell with pumps. So the calcium levels initially increase, shutting off the burst, but then other mechanisms regulate the level of calcium, so they decrease it, removing the inhibition, allowing the calcium influx to occur, and the cycle repeats itself over and over and over again. So what we see here at the nano network level is really just a sort of recapitulation of a feedback inhibitory network. One cell connects to another, that cell then leads to activation of an inhibitory process which shuts it off. So conceptually, this nanocircuit within the neuron is very similar to a microneural network. And this theme, I hope to show you, is repeated over and over again. So some of the same motifs that are used in neural networks are also used for subcellular phenomena. Let me move to a more complex kind of rhythm, and that is the kinds of rhythms underlying locomotive behavior, and particularly talk about quadrupedal locomotion because it's more interesting and complex, because quadrupeds are capable of not only moving their four legs, but generating different types of cycles of activity. Shown here is a walk in which there's a cyclical pattern. First, if you want to start with the left front, after the left front limb moves, then the right hind will move. And then next comes the right front, then the left front, and then we go back. I say left hind, and then we go back to the left front. Another gait is the trot, where we have the left front and right hind in phase with each other, and 100 degrees out of phase with the right front and right hind. Another example of a quadrupedal gait is the bound, where the left front and right front are in phase and 180 degrees out of phase with the left hind and rear hind. And then the gallop, which is a variant of the bound in which there's a slight phase difference between the right and left front and rear limbs. Now, how is it possible that the nervous system can generate these gaits? Does one need, for example, 4 different neural circuits to switch between one gait and another, or can one neural circuit do it all? Well, I must say in advance we don't know the answer to that, but there is some evidence pointing to the fact that specific circuits are involved. One way to do it, I should point out, preface it by saying, would be to take 4 neurons, individual neurons, and assign activity in each one of these neurons to move a limb. So you can have this neuron be a motor neuron that would move the left front. You can have endow this neuron with an oscillatory behavior, like I've already shown exist. You could have a second neuron that's involved in controlling, and of course this is a great oversimplification, but in principle you could do it this way. You could have another neuron involved in moving the left hind limb, a third neuron involved in moving the right hind limb, and a fourth neuron involved in moving the right front, right? And endow each of these neurons with its endogenous properties. So in principle this could work, but you would have some severe constraints because you would have to, and here you see the phase relationships that might be appropriate for a gallop. So these guys would go first, for a bound rather, and then be followed by these rear ones. But you would have to start these guys off at exactly the right time. And with time, there's the possibility that there could be some drift in the period. So what you might wind up is something like this. So this horse is not going to win any races, and it's probably not even going to be able to walk. Now, one way of doing this is to use a network. Again, we don't know the details, but what seems to be important in these kinds of networks that generate locomotive behavior is reciprocal inhibition. And a specific implementation of that is this recurrent inhibitory circuit or ring circuit. This is actually work that was done by graduate students, Carmen Canavia, Rob Butera, Ron Doerr, working with Doug Baxter and me and John Clark in the electrical engineering department here. So what we did was to design a circuit like this, a so-called ring circuit, where each neuron has within it an oscillatory behavior, and each neuron is connected to another neuron through a synaptic connection, using biologically realistic neurons and biologically realistic synaptic connections. Now, in order to get the phase relationships right, rather than assigning neuron 3 to be the right front, as it would be sort of indicated here, we have to assign neuron 3 to be the right hind limb and neuron 4 to be the right front limb. And that puts things in the right phase relationship. Now, when you do this and you implement this circuit, single circuit with just small parameter changes is capable of generating these different gates. So this indicates, I think, an important point about neural networks is that neural networks, you need to understand not just the topology of the network, but you need to understand the nature of the connections between the neurons, whether they're excitatory, whether they're inhibitory, and the nature of the way in which the nodes work. Here we have oscillatory nodes. So it's both the properties of the nodes and the properties of the connectivity that make this work. And also this illustrates a phenomenon which is called as dynamic reconfiguration. So one network, you don't need multiple networks to generate these 4 different gates. One network can do it. It can be reconfigured from generating a walk to generating a gallop just by slight adjustments in the parameters of the synaptic connections or in the properties of the oscillatory cells. Let me just briefly talk about a rhythm which is very prevalent in our daily lives, and that's the circadian rhythm. Circadian rhythm is due to the operation of a group of cells in a region of the brain called the suprachiasmatic nucleus, located just underneath the optic nerve. And those neurons have profound effects on both hormonal release, such as melatonin and cortisol, as well as on autonomic functions, such as body temperature. Now, despite the fact that so many physiological systems are affected by this, the clock mechanism is quite simple. Here's a little schematic diagram of the basic components. We have a basic oscillatory mechanism, or the so-called time-keeping mechanism, the core clock. That clock can be set or reset by light and temperature, for example, and then that clock produces output to regulate these other behaviors like locomotor behavior, the sleep-wake cycle, and body temperature. Now, despite the profound effects of this oscillator, this boils down to a very simple network, and indeed not even a neural network, another one of these biochemical genetic networks. And this mechanism seems to be conserved across all animal species, including man. Basically, it works like this. It's a genetic regulatory network, and it involves, in particular, a number of genes, but one that seems to be particularly important is this so-called PER gene. PER is for period. This gene was first identified from Drosophila, but is also present in vertebrates. So the PER gene is in the nucleus, and the PER gene leads to the production of PER messenger RNA. The PER messenger RNA then leaves the nucleus and goes into the cytoplasm, where it leads to the synthesis of the PER protein. Now, what does the PER protein do? It diffuses or is transported back into the nucleus, where it represses the further transcription of the PER gene. So this is one of these systems which gets activated, and then it gets inactivated. Conceptually, you see, it's very similar to that bursting neuron mechanism. The gene does something, it produces the message, produces the protein, which feeds back to inhibit the gene expression. Now you'd think, okay, you're only going to get one cycle out of the system. Well, what happens is that this per protein is degraded, and it's degraded over a 24-hour period. So as the per protein is degraded, the inhibition or the repression is removed, allowing this gene to start making messenger RNA and protein all over again. So once you get this cycle going, it repeats itself over and over again at this 24-hour period. So this is the core mechanism underlying circadian rhythms and the powerful effects that they have on a number of different physiological systems. It's a feedback or recurrent inhibition network, basically. Now let me turn to, in the final moments here, of something that's dear to my heart, the neural circuits for memory. I think it's fair to say that memory is obviously a critical phenomenon for all of us. It makes us what we are, and without it, life would have little meaning. There's been tremendous progress in understanding the neural and molecular mechanisms for memory over the past 30 years. We know regions of the brain that are important for memory storage, and we know from the work of Eric Kandel and others much about the molecular mechanisms for memory. We know that memory involves changes in the strength of synaptic connections. So we talked about synaptic connections, and we've sort of inferred, and many people think, that they are like solder joints between elements of electric circuit that are rigid and fixed. But in reality, the strength of a synapse can change, and it can change as a result of experience and as a result of learning. So learning involves changes in the strength of existing synaptic connections within neural networks. To learn, when we learn and remember something, we don't have to construct a new neural network for the memory. The memory is stored within an existing neural network. We also know that long-term memories, the kinds of memories that last days, weeks, and lifetimes, involves changes in gene expression and protein synthesis, whereas short-term memories do not. Now, what is limiting in the understanding of memory is the details of the complex memory circuits in the brain. We know regions of the brain that are important for learning, like the hippocampus and the temporal lobes and frontal lobes, but the detailed circuitry is a bit elusive. But we do have some glimmers of important neural circuitry that might be involved in memory. And one circuit that seems to be important is this recurrent excitation circuit. A specific implementation and example of this circuit and how it might be involved in memory is found within the so-called CA3 region of the hippocampus. And here's a cartoon of that circuitry. Now this looks very complex, but hopefully I can pare it down to size so we can see how this circuit works. So here we have 6 different hippocampal neurons, so-called pyramidal neurons, and I give them these little triangle shapes. So there's neuron U, V, W, X, Y, and Z. Right? Each one of these neurons receives a synaptic connection from these presynaptic neurons. This one is labeled A, B, C, D, E, and F. Right? These presynaptic neurons can be either active or inactive. So an active neuron is labeled green, and an inactive neuron is labeled black. An important aspect of this circuitry is that this synaptic connection is sufficiently strong, like this synaptic connection A is sufficiently strong to activate neuron Z. So if neuron A is active, neuron Z will be active, and there will be an output at the axon of this cell, indicated here by this green 1. So this is nothing more, really, when you look at it as feed-forward excitation. Now, what's special about this circuit, however, is that neuron Z and all the other neurons make so-called axon collateral connections, pathways, to feedback and connect to themselves. So for example, this Z cell comes around and makes a synaptic connection with itself. And it not only makes a synaptic connection with itself, but it makes a synaptic connection with the 5 other neurons in the circuit. Now, just as cell Z makes a synaptic connection with itself, so does cell Y, so does cell X, and all the other cells. So every cell, cell Z, for example, is receiving convergent information from all the other cells in the network, and in turn it is diverging to make synaptic connections to all the other elements in the circuit. So we have a matrix of connectivity here. This is called, by the way, for reasons that will become clear, an auto-association network. Now, in order to have this network learn anything, we have to embed in the circuit a synaptic plasticity learning rule. And the rule that we're going to use, and this is the physiological rule that's been discovered, is a rule that's known as the Hebb learning rule. And essentially it states that a synapse, given by one of these little triangles here, will change its strength if that synapse is active, releases transmitter, at the same time the postsynaptic cell is active. So if the postsynaptic cell, let me just say, if the postsynaptic cell is active, cell Z is active, and the presynaptic cell is active, this synapse will change its strength. Now this synapse is going to change its strength quite readily, because if input A comes in, it will activate cell Z, and cell Z will come around and cause the release of transmitter. So as a result of activation of cell A and its consequent activation of cell Z, this synapse will be strengthened. But this is not the only synapse that will be strengthened. This synapse will also be strengthened. Because cell Z was active, it will release transmitter at this synapse 13 onto cell X, but cell X was also activated by this other pathway. So this synapse will be strengthened by a combination of activation of cell A and cell C. This synapse will not be active, even though this synapse will not be plastic. It's releasing transmitter, but the postsynaptic cell Y is not being activated. So the net effect, because of this convergence and divergence and this plasticity learning rule, an initial input pathway such as this will be stored in the matrix of connectivity. An important concept here is that the memory is not in any one synapse. It's distributed in this network of synaptic connectivity. So you can think of this as a matrix, a connectivity matrix, and there's either, you can view these as ones and zeros, a one being a learned synapse and a zero being an unlearned synapse, and the memory is that matrix. So the memory then can be stored in the synapse, but how does that memory persist? So for days, weeks, and lifetimes. Well, here we have to then turn to a biochemical and genetic network. And the next slide illustrates a particular network that we've been working on in my lab, and Eric Kandel and his colleagues as well. And this is the network that allows that long-term switch to take place and be maintained. Now, there's going to be a quiz afterwards, so you have to remember all these pathways. The intent here is not to snow you with this complexity, but it is to illustrate that the biochemical networks that are underlying that plasticity are relatively complex. But despite the complexity, you see motifs in here. For example, you see a feed-forward excitation motif. You see a feedback excitation, a recurrent excitation motif. And you see a feedback or recurrent inhibition motif, where this gene which is expressed feeds back to repress its further expression. So we see these motifs over and over again, the same kinds of motifs you see in neural networks, you see in biochemical and genetic networks. So to summarize, I've talked about a number of different network motifs. We've made considerable progress, neuroscience as a field, in understanding how different simple neural networks are involved in mediating reflex behaviors utilizing feed-forward excitation, feed-forward inhibition. We've seen how lateral inhibition is important for contrast enhancement. We've seen how recurrent excitation can be an important mechanism for memory circuit, how recurrent inhibition can be important for generating locomotor behavior, and these same kinds of network motifs are recapitulated in cellular molecular mechanisms as well. And perhaps we should turn it around that the neural networks have learned from the cellular motifs because the cellular motifs were there before the networks. Now, despite all the progress that's been made, we're still just beginning to skim the surface in understanding more complex, so-called higher order functions of the brain, such as those involved in cognition. And as one example of cognition, take object recognition. So here's an image that you perhaps recognize as a portrait of a general. The question is, what kind of neural networks are involved in that object recognition and giving that object meaning? Why is it that, well, some of you see a general, others may see lost my mouse, others may see a dog and a man and a woman. And others may see both the general and the man and the woman, but probably not the two at the same time. So we really, these are challenges for the cognitive neuroscientists that are here, Jim and Suzanne, to figure out the neural networks that mediate these complex cognitive phenomena like object recognition. Much progress is being made, and more will be made. We are beginning to understand the areas of the brain that are important for these kinds of phenomena, like object recognition involving the occipital cortex and temporal lobes, using both recording techniques and modern imaging techniques such as fMRI, functional magnetic resonance imaging. So we're beginning to understand some of the modules, the brain areas and the modules that are important. This one, there was an article in the student newspaper, and the title of my talk by the students is, "It's a rat's nest of wires up there." Well, here's the rat's nest right here. This is from my colleague Dan Feldman in the department. It's an old slide, and there's been some further developments here, but conceptually, it's the same picture. Processing, it starts down here in the retina, and then multiple cortical regions are involved, the occipital cortex, the temporal cortex, and within those cortical regions are the so-called modules that are extracting higher order information from these visual scenes. A challenge for the future is to determine exactly how these modules are working and how they're interacting with other modules. It's not clearly a simple hierarchical scheme. There is feed-forward connections, admittedly, but there's also feedback connections and lateral connections. Now, what we've learned from the analysis of the simple networks is that in order to understand them, you need to know more than the topology of the network interconnections. You need to know about the dynamics of those connections and their signs, whether they're excitatory and inhibitory, and you need to know about the ways in which the nodes work. And these nodes here, each one of these nodes presumably involves hundreds, if not thousands, of those individual microcircuits. But with further research, hopefully we'll understand these nodes better and the networks that interconnect them. So ultimately, we'll be able to understand better this node and how this node connects to knowledge and societal networks. So I'll stop there, and thank you very much.
Questions if you have them, and he will just repeat the questions so that our video recording picks it up.
Yes, ma'am.
Could you take us through the path to the garbage pail in the earlier slide about the retention of memory?
Could I take you to the path?
Well,
Oh, the one with the garbage pail?
there were a couple of garbage pails, which I assume means memory is...
Oh, the garbage pails meant degradation, protein degradation. Yeah, so yeah, that's not the memory going in the garbage right there. No, that's degradation of this particular messenger RNA.
Okay.
So we have, throughout this process, we have a balance of synthesis and degradation. And so an interesting conceptual issue is that how do you maintain a long-term memory, especially one that lasts for weeks and lifetimes, in the constant presence of protein degradation? So it's easy to think of a memory that could last a day or so with new protein synthesis, but how do you keep that protein level increased? Well, you do it through these positive feedback networks. So once you get the system going, the system, and this kind of a positive feedback circuit over here, I won't go into the details of it, but there's multiple positive feedback micro networks within this network that keep the biochemistry and the protein synthesis going. Yes, sir.
So you've essentially modeled the brain as a network of digital circuits, digital gates. And since the brain doesn't contain very many more of these gates than a modern microprocessor, where does the power of the brain come from? The brain obviously is much more powerful than my desktop computer.
Well, some are, maybe. Well, I think the key is perhaps here in this connectivity matrix. Because just think, so you have 100 billion neurons. Now, if you'd like, and this would be an oversimplification, you can think of each neuron as a bit. Either it has an action potential or not an action potential, right? That could be the memory. But each neuron gets 10,000 synaptic connections. So, and makes 10,000 synaptic connections. So each one of these synaptic connections is a potential bit of information. So this synapse can be either modified or not modified. So it's not just, if you take 100 billion neurons times 10,000, then you start to get into a number, I think, that might exceed your personal computer. And in addition, it's just not ones and zeros. You know, I've portrayed it that way, but the synaptic weight just doesn't change from 0 to 1. It's actually a continuous process. So you can go from a synaptic potential, which is 2 millivolts, to 3 millivolts, or 4 millivolts, or 8 millivolts. So how that information is encoded, we don't really know. So it's not just a digital computer. It's a combination of a digital and analog computer. And we also don't know for sure what is the real computational element. I mean, because all that biochemistry that's going on in there too could be important that we don't appreciate. We think that it's, you know, maintaining the network structure, but maybe it's doing other things. So maybe part of the computation. Yes?
Yes, it seems to me that as I get older, the problem is not memory, the problem is recalling. So do you have any ideas about how things are recalled from?
Well, this is a very difficult question. So memory can be divided into 3 phases. There's the acquisition of the memory, and that's getting the information in there. Then there's the storage of the memory, and then there's the recall of the memory. So it's hard to, when you can't recall a memory, it's hard to know whether the problem is in the recall mechanism or in the storage mechanism. So, and there's a big debate about that. Now, I think it's clear that some memories do fade away. But other memories that we think fade away, we've all had this experience. You try to remember the name of your kindergarten teacher and you can't do it for the life of you. Then 2 days later, it pops into your head. And so it's not that it wasn't there, it's that it was there, you just couldn't access it. Now, one of the big mysteries of memory research is how exactly that recall process works. And it's very difficult to separate a problem with recall and a problem with storage. Would you agree, Jim?
Yeah. I had a better answer, but I forgot.
Okay. Yes, Suzanne?
I'm thinking about the difference between humans and other brain-having organisms. So the architecture that you showed, that array of different kinds of mechanisms that could be then made into higher order processes, where do you see the difference between a human brain and, let's say, other mammalian brains or other brains of other species? Is it surely in the number of neurons? Is it in the different kinds of loops and feedbacks that you get? Or where does it reside?
Well, in terms of the architecture, it doesn't seem as though there's any fundamental difference between the cortical architecture of a human, a monkey, a cat, or a rat. I mean, the same kinds of, there's no special neurons, there doesn't seem to be any kind of special synaptic connections, and there doesn't seem to be any special patterns of connections. So it seems to be a numbers game. And these areas of the cortex that are involved in these higher cognitive functions are expanded in monkeys and humans. Monkeys have very similar cortical processing areas as do humans. So I think it's a numbers game. I'd like to hear any other opinions on that, but I don't know. Whether there's something that emerges out of networks when they reach a certain scale, you know, that's an issue that is a possibility. Yes?
There are definitely animals with like larger brains than humans, though, with that.
There are animals with larger brains than humans. I believe so, but it depends upon what they do. You can have an area of the brain, like I understand that some skates, for example, have very huge cerebellums. So why they have it, don't know, but that's not going to probably help their cognitive functions. Yes?
What's the status of research, say, insect brain, which is what can potentially be simple?
What's the status of research on insect brains? Well, insect brains, there's a couple of insects that are being used as model systems to study memory mechanisms, specifically the honeybee and the fruit fly, Drosophila. And there's been lots of progress made in those systems. Drosophila has been particularly advantageous because of the ability to do genetic manipulations. But the problem with the insect brains is that they're very inaccessible. So while you would think that they have simple nervous systems, they have complicated nervous systems. And the cells are very small, so it's very difficult to do the cell biology on an insect brain. It's very difficult to find. My own research and that of Eric Kandel's is on the sea slugs. We use those because they have simple nervous systems and large cells, so it's great for doing cell biology. But obviously a sea snail can't remember its mother's face, so it's useful for examining simple forms of learning like Pavlovian conditioning and operant conditioning, but it's not good for higher order features of memory and cognition.
Are there any other questions?
Jim?
You led off your talk by reminding us of all the wonderful things that take place between the two ears. Everything from hearing music to literature to perception and so forth. If you look across the cortex, where many of these tens of millions of cells are, although there are differences going across the cortical surface, I'm more struck by the similarities. That basically, the cortex is the cortex, whether it's in the part of the brain doing olfaction or the part of the brain doing hearing or memory or language. So do you think ultimately it will turn out that there are different networks that, say, allow us to smell an odor from hear a concert, from remember our kindergarten teacher?
The question is, how is it that the brain does so many different things using what appears to be the same kind of network architecture? That's a good question. I don't know the answer. It will be important, I think, to see if we can find some motifs for these, higher cognitive functions, percepts. Maybe there's motifs for different percepts, and they then. The problem is we don't know what the output is, right?
Well, if there aren't any other questions, I would just like to thank Jack Byrne for coming and giving this lecture and giving us an example of how such an extraordinary complex thing is built up from these simple sort of repeating units piled on in intricate ways. And the idea of memory as a matrix is one that's going to stay with me for a long time because I would not have thought of that. And so thank you all very much for coming, and I hope you will join us outside for a reception, speak some more with Dr. Byrne if you so care to do, with your colleagues, and join us next month when we are going to have Carrie Ward speaking about networks of empire from the history department. So thank you all very much, and thank you.
Thank you. Thank you.
That's fantastic. Thank you so much.
Oh, thank you.
This program is protected by a copyright and may not be redistributed in whole or in part without the express written consent of Rice Digital Media Services.